Hello friends and welcome back to another video. In this video that we will be talking about the topic antigens. Antigens are those substances those having a capacity for producing antibodies once it encounter with a particular person immune system is what we called as antigen. Antigens means these are foreign substances. Once it entered into a human body, they having a capacity for generating antibodies is what we termed as antigens. Not all antigen having a capacity for generating antibodies. Some antigens does not have the capacity for generating antibodies, but they are effectively takes place in cell mediated immunity as well as immunological tolerance reaction. If a specific antigen enters to a person, then the corresponding antibody will be produced in a particular person's body, and these antibodies will be complementary to the antigen that encountered to that person, which means here the antigen and antibodies are complementary with each other, which means that here antigen that possesses a small region that is called as epitope, which means a specific antigenic determining site, and antibody container the same small area as what we call as paratope, and this paratope and epitope can effectively bind and are capable for producing immunological reactions. A complete antigen are those antigen that are having a capacity for the generating antibodies, but there are certain substances which called as haptens. These haptens are not able to generate or not able for inducing the production of antibodies, but it can effectively react with antibodies. If the haptens has been bind with certain large carrier molecules, then it become immunogenic. So the haptens are mainly are of two type, that is simple haptens and complex haptens. Complex haptens are polyvalent and complex haptens are capable for precipitating, and simple haptens are not polyvalent and it is non-precipitating. Since it's assumed to be for the precipitation, the antigen should possess more than two or more antibody combination sites must be required. Then moving on to our next small topic, that is the determinants of antigenicity. And among the first one that comes under the determinants of antigenicity is the size. The size of antigen having a great relationship regarding its antigenicity, which means if the antigen possesses a large molecular mass, which means that are more antigenic, and if the antigen that possesses very low molecular mass, which means that are non-antigenic. And the next comes our chemical nature. Usually antigens are polysaccharide or protein, which means these antigens are more antigenic, and certain antigens compositions are lipids and nucleic acid, and those composition antigens are non-antigenic or less antigenic. And the next comes foreigners. In a normal person or the person who is free from certain kind of conditions are usually capable for distinguishing between the self antigen and the foreign antigen. Usually our body never produce any kind of immunological reactions against the self antigen, which means our body having a clear distinction between the self antigen and the foreign antigen. Our body produce antibodies only to against the foreign substances or foreign antigen, and our body never produce any kind of antibody to the self antigen unless a person is free from autoimmune conditions. And the next comes as species specificity. Species specificity means those individuals that contain certain antigens on only belonging to a specific species is what we called as species specificity, and they're always having a chance for the cross reactions to be occur between the antigens of closely related species. And the next comes as IsoSpecificity. IsoSpecificity means those antigens will not be present in every individuals, those who are belonging to a same species. The best example for that is human RBC antigens. This particular antigens is only present in certain individuals of a species. That's the reason why we having different blood groups. And the next comes as AutoSpecificity, that we already know that the self antigens will not accelerate the production of antibodies, which means the self antigens are normally they are non-antigenic. So our body will not produce antibodies against to the self antigen unless a person is free from autoimmune conditions. Next comes as organ specificity, which means certain antigens are present on organs such as kidney, lungs, brain, and all. Usually occur a complication if they're having any kind of brain damages, usually administered for anti-rabic immunization. They always having a cross reaction between the antigens that are present for between the humans and the sheep. So that's all about our topic antigen. And this was a very brief introduction regarding the antigen and the determinants of antigenicity. I hope you enjoyed my class. Thanks for your watching and please subscribe on my channel. Don't forget to smash the like button. See you soon and have a great day, guys.